Okay, folks. Today I'm going to talk about management of dark rover ants using baits in California. So I'm sure some of you are dealing with dark rover ant, and even if not, some of the basic principles are the same. Okay, because I'm going to talk about baiting, and I'm going to, talk about some very popular products that are commercially available to us for ant control, including Argentine ant control, odorous house ant control, and other ants. So the dark rover ant is a very small ant in the ant world. So it's about 1 to 2 millimeters, which is about 132 to 116 of an inch. So for comparison, Argentine ants are uh, 2 millimeters, 2 to 3 millimeters, or 116 to 3/32 of an inch. It's an invasive species, so it's not native to California. So anytime you see something in red, it's going to be useful for your quiz. So it was first found in California in 2010, but we don't know exactly when it arrived in California. So now you can find it in Southern California and lower parts of the Central Valley. So it's definitely in Kern County, and so it's in like lower parts of the Central Valley. You can find it in ag fields. That's what I've been hearing from my colleagues. It's not necessarily causing big problems there. And in urban areas, it can cause problems when they get into houses, when they swarm, or when they find their way into pantries and places like that. So you can see a trail of dark rover ant right here. As you can see, their trails are loose. It's not packed with ants, and they come in waves. You see like 5, 10 ants, following each other, and then there's a gap in between. So they form low density trails with gaps in between. So remember that. Yeah, so this one is actually from my own home. I have a beehive, and all these ants were going for wax and stuff. So the honey bee colony was collapsed. So yeah, there was a lot of honey and wax in the beehive. So one interesting thing about this ant is that there is no aggression between the dark rover ant and the Argentine ants or the red imported fire ants. So usually these ants, fight fiercely with other species, but you don't see anything like that between the dark rover ant and these two ants. But they fight fiercely among themselves, and they don't tolerate each other. So you can have one colony right here, and the next colony could be like a few feet away, and still they fight big time. So you can see these two I collected from the same place, almost from the exact same spot, but apparently they were from different colonies, and I had a hard time collecting ants and keeping them alive for my experiments because I didn't know that it's that bad, the aggression among these dark rover ants. So, but with other species, Argentine ants and fire ants, it seems like almost, they don't mind them having dark rover ants around. All right. So yeah, I mean, uh, the main reason that you don't see similar aggression in Argentine ants and red imported fire ants is that they form super colonies. So you can collect some ants here and then go several miles or sometimes many miles to one side and collect some ants there and then put them in a jar. You don't see any fight. This is a complete opposite of those polygyne colonies or that forms super colonies. All right. Okay. So what do they eat? They're scavengers. They feed on dead insect arthropods. You can see one dark rover ant here in this clip that is carrying the pronotum of some sort of insect. And I've seen them collecting tiny, tiny spiders. And of course, they love sweet stuff. So they're omnivores, meaning that they feed on both plant and animal matter. But they definitely like sweet stuff, honey, sugar, honeydew, and you see them going after these resources. So in the laboratory, studies have shown that they like carbohydrates, and I've seen it in my experiments too. They take honey water, sugar water, fine, and some seasonality was observed. So in the winter time, they went for more protein, and I'm sorry, in the springtime they went for more protein, and in the winter they went mostly for sweet stuff. And this is a typical behavior of ants. In the springtime, they're reproducing, so they need more protein to create larvae and feed them so that they can grow and become adults. But in the winter, the reproduction usually is it's not active, so they just mostly go after sweet sources, sugary sources. Okay, yeah. So I mentioned honeydew. Originally, it is known that these ants go after root-feeding hemipterans in their native range. But when they move, when they become invasive, they're very opportunistic. They go after many sources of food. Yeah, usually see them on tree trunks. And if you look at the lower picture here, you see that one of them is feeding on water droplets in a vial. And when they feed on stuff, their abdomen gets expanded, and you can see the space between the sclerites on their abdomen. So they don't build mounds. Okay, so unlike the especially the fire ants, they don't build any mounds, and all is visible outside on their nest is a tiny hole in the ground, and that's why sometimes it's really hard to find their nest. So you see them around, walking around, forming a trail, but you don't necessarily see or can detect the exact nest entrance. And of course, finding the entrance may not necessarily be enough because we don't know how deep, or like if it goes sideways. So it's a little bit tricky. One interesting thing behavior about this ant, some other ants too, is that when they find a food source, at least this was observed in the lab, they readily move their colony closer to the food source. So they don't like to form super long trails, but they move them, at least part of the colony, close to the food source, and then go back and forth between that, kind of like satellite colony, if you will. So this was observed in the laboratory. So now I'm going to talk a little bit about some general biology of ants, because there's some similar patterns that can be grouped together, and it's very useful not just for the dark rover ant, but for any type of ant that you're dealing with. So in many ant species, one interesting behavior is that only less than 20% of the colonies forage. So whatever you see on the ground, that's only a small fraction of the colony, okay? And this has very important uh, implications for the management of ants. Why? Because if you're targeting only the foragers, you're only targeting 20% of the colony, right? So guess what? Even if you kill all of them, there's always more in the ground, in the nest, and of course the larvae. So for example, it was found that a harvester ant colony had about 7 to 15% of the foragers, like is made of 7 to 15% foragers, meaning that the rest of the colony, 85% or more, they're not foraging. They're just in the ground, in the nest. And another study showed that for another species that only 10% of them are foraging, and 90% of the colony is not even on the surface. So they did a study in the lab, and they, as you can see, they marked all these ants, with tiny dots of paint, and they found that about 40% of the colony is inactive. So they're absolutely doing nothing. So when we talk about ants, the first thing that comes to our mind is that ants are hardworking, they're working all the time, but guess what? They're not. 40% of the colony is inactive in this species. Okay, so they're not tending the young, the larvae, they're not collecting food, they're not grooming, they're not cleaning the nest, absolutely doing nothing. So the researchers thought that these inactive ants doing absolutely nothing are served as a reserve labor force. So anytime you start killing these ants, what happens is that that reserve labor force all of a sudden becomes active and start to kind of like replace the dead population of foraging ants and continue collecting food and making sure that the colony has enough food to survive and reproduce. So this is very interesting, and that it really shows the importance of targeting the whole colony and not just the foragers, because again, look, about 10%, between 7 to 15% of the colony forages. That's it. The rest of them are on the ground. All right. Uh, so another interesting thing is necrophoresis. So it's a behavior of moving the dead ants outside, and termites do that too. So anytime workers see a dead ant, what they do is that they move the dead body out, and that's very important because it prevents diseases from spreading to the rest of the colony. And you see the same pattern when you use baiting or even like liquid insecticides. Let's say you use fipronil, and some of the ants, will get exposed to the fipronil or the bait, feed on the bait, go back to the nest, and they start feeling, weird, and then they start dying. And then as soon as they die, you see ants coming out and moving these dead bodies. So it's a sanitation practice, and necro means death related, and phoresis means a transport. So while this behavior is thought to be a way of kind of like protecting the colony, it can be useful for baiting and even like with liquid insecticides, because anytime ants touch each other or try to carry each other out of the move other members of the colony outside, by touching the dead ant, they're going to get exposed to more pesticide. So it's actually, research has shown that it's actually beneficial, and it helps to kind of like increase the impact of pesticide on the colony. So you see these graveyards outside colonies. I've seen this a lot with the Argentine ants and with the red and porter fire ants. So let's say you apply bait today, and in 1 week you come back and you see a lot of dead ants right outside the entrance of the nest. And again, it's a sanitation practice, but when you use contact insecticide, especially, it really improves the efficacy of pesticide, because all those workers carrying dead bodies will get exposed to pesticide, and they're going to be the next dead ant. So when we talk about ant control, especially, you may hear a phrase that is horizontal transfer. So what does it mean? It means the exact same thing that I just talked about. So ants feed on a bait, right? And they go back to the nest, and then they exchange food, like you can see here, right? And the food that they're exchanging has a toxicant in it, right? Because it's a bait. And what happens is that the second ant gets some dose of that toxicant or insecticide, and she's gonna die, right? And if there's enough time, the second ant or the recipient of the bait may have enough time to go and pass the toxicant to another one. So one ant can, depending on the situation, depending on the species, depending on the toxicant, can kind of like pass the toxicants to 5, 10, or 20. So this is a very good, this is very beneficial for any baiting program, and we call this a horizontal transfer. But horizontal transfer, as I mentioned, doesn't have to be through baiting, through exchange of food, but anytime they touch, with their antennae, with their legs, that can also expose other ants to the same toxicants. So it can really increase the impact of insecticide on the colony. So as I said, you have trophallaxis, which is the technical term for exchanging food mouth to mouth. And then you have the contact, let's say you put any type of fipronil, it can really spread to the colony by just contacting, through the antennae and legs. And just as a reminder, not all pesticides have the same level of transferability. So fipronil, generally speaking, has a very, very high transferability like property. So it easily gets transferred by contact between ants, and that's one of the reasons that fipronil is so effective. But of course, because of the regulatory concerns, more and more pressure is being put on fipronil, but it has excellent transferability. All right. So now talking about the speed of control. And again, this applies to all ants. The slower the chemical works, the higher the horizontal transfer rate is going to be, because there's more time for the exposed ants to go back to the colony, exchange food, or just contact each other, touch each other, and then pass the toxicants to the rest of the colony. But if workers die fast, you're going to have a less chance of transferring the insecticide to the rest of the colony. And that's why most effective products for ants have a slower effect. They're slow acting insecticides. Okay. Sometimes, I'm not against using fast acting insecticides, because in some situations, it totally makes sense to use a fast acting one, especially if you have an emergency and all you care is just to get rid of foragers. But as I said, only a small percentage of the colony forage, excuse me, and even though you're killing all the foragers and things look really good initially, the colony can easily come back and kind of like revive, or just send their like inactive workers to do more foraging after a few days. So as I mentioned, fipronil is readily transferable from ant to ant, but bifenthrin and some other pyrethroids are not good in that. So again, you can use them as repellent, as a barrier, but generally speaking, we should not expect to eradicate or have a huge impact on the whole colony when we're using pyrethroids. All right. So another interesting thing about ant control when you're using baiting is that the larger the colony, the higher amount of insecticide is needed to control the colony. Okay. So if you're using very low percentage of a bait, let's say 1% boric acid, it may work for smaller colonies, but when you think about it, when you're dealing with a super large colony that is made up of many thousands, many hundred thousands of ants, the toxin, the 1% boric acid toxin that you're providing to foragers is going to be passed to many, many, many other workers, and it's going to get more and more diluted. Okay, so it quickly loses its efficacy when it gets diluted that much. So the ants that originally feed on the toxicants, we call them donor ants. So the donor ants are going to be exposed to a much higher dosage and or concentration of the toxicants compared to the next ones, and then the next one. So a percentage or a concentration that may work on smaller colonies may not work on a large colony. So please bear that in mind, especially if you're making your own concentration, because some of the baits allow you to do that. Like for example, taro bait. You can buy a jug, and I think it's like it comes in a as a 5% concentration, and you can bring it down to 2.5% and 1%. So if you're dealing with a very large colony, my suggestion is to go with a little bit higher. Don't do like 1%. Go by maybe 2.5% or even like 4 or 5%. Sometimes they work fine. But again, if you use the 5%, you're going to kill foragers more than when you use a 1% concentration. So that's something, another thing to keep in mind. Just to review about what I said in the last several slides, most of the workers don't forage, but they can be recruited anytime needed. So we call them inactive workers or workers that they don't work at all. So they're just there ready to be recruited, especially when a lot of them die all of a sudden because of insecticide applications or baiting. Yes, so killing foragers, you may get quick results, but you're only killing a small percentage, like less than 25% of the colony, and those dead ones will be replaced. So if you give them enough time to any colony, and when you're doing baiting, you're going to have a higher impact on the colony. But it just needs more patience, and the technique and the whole process needs to be communicated to your clientele, to your client, to your customer. Because if the customer doesn't understand why you're using a low percentage bait or a slow acting bait, they're not going to be happy with your service, right? So that definitely needs to be communicated to the customer very clearly. And if they're open to it, you're going to have a higher impact on the colony, and then you're going to have much better results in the long run, versus using a fast-acting bait. So I talked about the dilution and the problem of dealing with very large colonies. So any toxicants you provide will get diluted, and it's not going to be super effective. All right. So as I was saying, faster control means lower chance of colony elimination, okay? At least most of the times. So usually we classify baits, especially as into like fast, medium, or slow acting active ingredients. So anytime you have a neurotoxin, something that has either some sort of pyrethroids or neonicotinoids, you're dealing with neurotoxins, and they usually act fast, within hours to maybe a day or two. Anytime you're dealing with a metabolism disruptor like hydromethanol that you can see in Amdro products, they're a little bit slower, and because the way they act is that they disrupt the energy production of ants, but it takes a little bit time, okay? And the slowest acting products are insect growth regulators, so they're going to have a major impact on the colony, but it takes a long time for them to start showing effects. So some of the examples with fire ant granular baits you can see here. So neurotoxins work in 2 to 7 days. Metabolism disruptors, they usually work in 2 to 6 weeks, and insect growth regulators require 6 to 8 weeks or more. So something to keep in mind, but definitely each one has an application. So depending on the scenario, depending on what type of pest you're dealing with and what your customer wants, you can use different ones. So I've used IGRs a lot side by side with neurotoxins, and although you get quick results with the neurotoxin, so which is really appealing because you go back to the exact location where you applied the bait, and you see absolutely zero foragers. So this is really appealing, but the problem is that they always come back quickly. So I had better results with IGRs in the long run, but I had to wait weeks and weeks, to see any effect. And that's why some of the products mix an IGR with maybe a metabolism disruptor. So you get faster suppression, but the long-term effect is going to be there as well. And you're going to see that after some weeks or maybe even months. All right, so I think I covered everything about these things. And yeah, so advantage of baiting versus chemical spray. So there's no risk of drift. If it's windy or there's a breeze going on, you don't need to be worried about having drift. So pesticides go from one property to the other one. So reduced risk of environmental contamination. So anytime you apply stuff to the on the ground, especially on hardscapes, like walkways, sidewalks, and driveways, and just on concrete around the house, on the perimeter of the house, you need to worry a little bit at least about pesticide runoff, because if it rains or if the irrigation kicks in at night, some of those pesticides are going to be washed and get into the turf or just go into the storm drain. But with baits, you don't have that concern for the most part. Um, and as I said, most baits are designed to work slow or relatively slow, and that gives you a chance to kill the queens and the brood. And this is really important because, as I said, we don't want to just kill the foragers. We definitely want to go after the whole colony, including the queens, brood, and those inactive foragers in the colony. All right, so now I'm going to talk about my actual experiments on the dark rover ant using baits, and I'm going to mention some of the products that I use. I'm sure many of you are familiar with some of these products. So it's interesting how different products work differently on different species. So there have been some previous studies on the dark rover ant. So you can see one here. They tested different products. They use siesta granule that is used primarily for fire ants. They use Terra PCO gel. And as you can see, PCO gel was the most effective one, and it has borax in it. So that was an interesting study. As you can see, it takes some time for borax to kick in. So it wasn't until the 11th day almost that the products start to kick in. So borax and boric acid can be very effective, but you definitely need to be more patient because you can get much faster results with other products and other active ingredients. So another study in 2014 in Arizona, they did a lab study and they used enticed rover ant bait, which has borax too. And they use Max Force Quantum ant bait, and they use also Advion. And you can see the results here. The only one that worked was the Max Force Quantum, which has imidacloprid inside. The other ones, this is the Entice with borax in it or sodium tetraborate. The indoxacarb also didn't work well, and the green one here is the control. So the only one that was significantly performing better was the imidacloprid application. However, I looked at their methodology, and the way they did it was they provided an alternative food source in their baiting arena. So the ants could feed on either the bait or something else. And apparently these some of these baits were not the most preferred food. And I found that that's probably the reason that they didn't get any good results, especially with the borax. But I'm going to elaborate, in the next slides. All right, so for my own experiment, I use a bunch of products from different companies, and that's what I really like to do. Not just try one, two products, but just try to expand my research into several products. So you have OptiGuard ant gel, you have Advion ant gel, you have Samaris ant gel, and this is the exact product that the previous study that I showed used Entice rover ant bait. So it's a bait that's specifically registered for rover ants. And this is the Max Force Quantum ant gel. So this is a very good product too. So all of them are good, but I just wanted to see which one is more effective against the dark rover ant in California. So I started collecting ants from the field. I went to different places in Riverside, Southern California, collected them. Initially, I was putting all the ants into one vial, but I learned it the hard way that when you do that, you're going to have a lot of aggression. And for example, if you collect 100 and in 24 hours, only 20 of them will be alive, because as I said, they just fight fiercely and they don't tolerate each other. So after like learning that, so I started collecting them separately from each spot. And I learned that when you provide a food source and they form a trail, that trail, if you collect from that trail, you're going to have a much pure, a more pure kind of like members of a single colony, versus if you're walking around and collecting ants, because when you do that, those ants could be easily from different colonies, and then you're going to run into the same problem of very high aggression and a lot of mortality in your vials. All right, so then I use paper towels, I put them in each vial, and I provided water and also the bait or sugar water for my experiment. And as you can see, when they feed a lot on liquids, their abdomen gets distended, and that's how you can see the space between these dark sclerites. And you can tell that this ant, has been feeding on liquids, either the bait or especially, like water in your vials. So that's how I did it. And I also use an ant nectar product that has sucrose in it, and I provide it in my control vials to be able to compare the effect of bait. And I put everything in like a centrifuge kind of like tray, and I had 5 replicates, and I had 10 ants in each vial, and I starved them for 12 hours. And this is done to make sure that the ants are going to feed on the actual bait quickly, because when you want to assume the efficacy of or judge the efficacy of products, you want to make sure that they're already impacted by the bait. And I put everything in an incubator and I monitored them. So here's the result of the first experiment. So I had a control, which is in blue here, and then I had Advion and gel. So similar to the Arizona study, I found that Advion is not an effective product, because I was providing Advion with water, but no other food source, and yet Advion failed to control anything. So it was very similar to the mortality of, or survival, I should say, of control. So this is a survival. This shows survival, and in the beginning you have 50 ants per treatment, all of them alive, and then as the experiment, progress, more and more died. So you can see that Max Force was the most effective product in killing dark rover ants. So in a matter of a day, half of the ants died in Max Force treatment. After that, OptiGuard did pretty well. And then you have Samari, which was good. Entice was the slowest one, as expected, because anytime you use borax or boric acid, you need to bear in mind that they're going to be slow. But all of them, killed the ants in about 7 days, or almost all of the ants in 7 days, and then 100% they caused 100% mortality in 8 days. So except Advion. So this is very similar to the previous study, but the Entice, which has borax, I definitely had a different experience with it, and that's what I said. That's why I said that I think that has to do with the methodology of my experiment, in my experiment versus the other experiment, because they provided an alternative food source, so probably the ants even didn't feed much on the borax bait. But in my case, it worked fine. You just need to be more patient with it. Okay, yeah, and these rectangles show similar products. So products that are in the same box, they're similar and there's no significant difference among them. But for example, this box and this box, they're significantly different because they're not similar basically. And these are all based on statistical analysis. So the experiment, the second one, was similar to the first one, but what I did not provide water to those baited vials. So all they could feed on was the actual bait and no extra water source. And if you look at the results, it's very interesting. Advion, which was not shown to be effective, all of a sudden started to kick in and kill the dark rover ants. So also Entice, which was one of the slowest acting uh, products, with borax in it, all of a sudden became almost the fastest product. And although there's no significant difference among these treatments, you can see that Entice really killed stuff faster than other treatments. So I was confused about the results. I was like, what's going on? I mean, Advil didn't work that time, and Entice with borax in it was very slow, and why it's so fast this time. So I realized that it probably has to do with the water stress, dehydration stress. So I did another experiment in which I used like a control with water, no water, and I had Entice with water and without water, and Advion the same, and Max Force. So just to see everything side by side to make sure that the difference that I saw in the second experiment has to do with dehydration. And I was right. If you look here, um, all these ones on the left and lower left, these are all without water. And Max Force, again, was the fastest acting. Entice was pretty good and kind of like fast. Advion was effective, but as soon as you look here, start from this one. So this is Max Force without any water. So you can see that they killed, or they were like a little bit slower compared to when there was no water in the vial. But Advion again was not effective against dark rover ants, and the mortality it caused was similar to the control. So I can be sure right now. I'm kind of sure that so Advion, for some reason they don't like it. I mean, I'm sure many other ants are like it, but dark rover ants for some reason they don't like this bait, but they definitely like Max Force, and Entice also works fine. Um, because sometimes like the dehydration effect that I studied, sometimes researchers want to look at why sometimes baits work well and sometimes they don't. And one of the factors that can affect the efficacy of a bait is the starvation. So if the ants are really starved, they're going to feed more on a bait and they're going to die faster. But if they're not starved, they already fed on various food sources, and all of a sudden you provide them the bait, uh, probably they feed on it a little bit, but you're not going to get the same results compared when you starve them. So you can see here, and this is from a research in 2012. So when they starved the ants for 24 hours, this is what they got. So these are the survival plots. So initially you have one, which is 100%. So 100% of them are alive in the beginning, and they start to die, all the way down to 0. So these two are from 24-hour starvations. But if you look at right, you see that when they starve them for 48 hours or 2 days, they got much faster results. So they reached 100% mortality in like about 6-7 days, and versus about 20 days when they starved them for 24 hours. So I was interested to look into that as well. All right. So as I said, they look into the starvation, and I was interested in doing the same thing. So what I did was, let me see. Okay, so I use several products, and actually I use one product. I'm sorry, this is probably not correct. So I use Entice for the most part, and then I had control, and I had different treatments with different hours of starvation. So if you look here, I had Entice. So this is my control. So I provided sucrose water, like, or sugary water, and I starved them for 24 hours. And as you can see, most of them stayed alive. I didn't have much mortality here. This one I had Entice, which has borax inside with water. So they had access with water, all of them, but I did not starve them. And as you can see, they kind of like start dying a little bit later. And then when I starved them for 24 hours, I got this line, and then the lower one in yellow was starved for 48 hours. But if you look at the results, you can see that these two are similar, and then these two are similar. So the only significant difference is between when I did not starve the ants versus 48 hours of starvation. So 24 hour starvation did not cause uh, any significant change in the efficacy of baits. So I think this is my last slide, but I want to show you something different here that I forgot to mention, I guess. Uh, yes. So what I was trying to say was that about these vials. Depending on what species you're trying to study, sometimes you can put them individually into vials. But anytime you're dealing with the social insects like ants and termites, you need to take into consideration that they may not behave the same way if when they're separate versus they're in a group. And that's why I decided to put them together, like 10 ants in each vial. And one question that is usually brought up in ant research and termite research is that, well, when you're separating ants or termites without their queen or without their broods, so how that experiment is going to translate into like into a field situation. So some studies have looked into that, and they have found that some species of ants, including the dark rover ant, behave very similarly in regards to foraging. So even if you take some of the foragers out and there's no queen and there's no brood, they still behave very similar to an ant colony that is outside in the field, or similar to an ant colony that is inside the lab, but with the queen and with the brood. So that's why in this case I don't believe that there's any like negative effect of taking them out, but it doesn't mean that everything is going to translate well into a field study or a field situation. So my next step of my research is going to be testing the same products in the field, because you want to make sure that what you find in the lab is going to be similar in the field. And so let's say you saw that how it happened, what happened when I had the water source inside vials versus when I removed that, and it totally changed the way uh, uh, ants basically survive. So I had much more mortality in the treatments that did not have water, and then um, the speed of control also was very different. So that's why it's really important to do field studies too. But doing lab studies is very helpful because in the field there's so many variables. And like we did some studies in the past, on Argentine ants, and anytime you go to the field, you see something new. Sometimes you get heavy rain. Sometimes your research site gets destroyed by heavy machinery, or the homeowner all of a sudden decides to put pavers on your research plot, or so many things happen, and you spend a lot of time in the field, you collect the data, but after 1 year, you look at your data and nothing is clear. Nothing is clear and necessarily significant. But in the lab, you can have much more control on those parameters, and then you can translate your findings and experiments and bring the whole thing to the field, and then do another test in the field. So I'm done with my talk. So if you need to contact me, please shoot me an email and I would respond